It's A. Sorry, never mind.
The clinical test used for the diagnosis of typhoid.
I put D. Yeah, D. Right.
Diseases, they are broadly grouped into infectious and non-infectious diseases. In the list given below, identify the infectious disease.
Fourth one is D again.
Which one?
D.
Correct. Influenza and the smallpox. Sporozoites that cause the infection when a female Anopheles mosquito bites a person are formed in.
I put C.
Wait. No, they're not formed in the salivary glands. They are present in the salivary glands. They are formed in the gut of the insect.
Oh, okay.
The disease chikungunya is transmitted by.
Huh? Sixth one I put as B.
Many diseases can be diagnosed by observing the symptoms in the patient. Which group of symptoms are indicative of pneumonia?
I put A.
Cancer-causing genes are called as.
C.
C, oncogenes. And if they are inactive, then what are they called?
I don't know.
The oncogenes. So next, in malignant tumors, the cells proliferate, they grow rapidly and move to the other parts to form. This stage is called as.
B.
Very good. When an apparently healthy person is diagnosed as unhealthy by a psychiatrist, the reason could be.
Question number C.
Which of the following are the reasons for rheumatoid arthritis?
D. First and second.
First and second.
I mean, first and third. First and third. D. Option D. I've put
The
D.
ability to differentiate pathogen or foreign molecules from cell increases. No, this doesn't. It decreases. The body
It
takes cells
decreases.
as the pathogen. The body is not able to differentiate.
Yeah, ability to differentiate decreases. Sorry, I read as decreases. Sorry.
So answer would be 2 and 4.
Yep.
Body is attacking itself, and then the ability to differentiate is lost. This one is correct. AIDS is caused by HIV. Among the following, which one is not a mode of transmission of HIV?
C.
SPAC is a drug obtained from.
I put A. Latex.
The substance produced by a cell in viral infection that can protect the other cell from the further infections are called as.
C.
Interferon. And which type of immunity does, like, is it? Is it the primary one or the secondary?
1 second, I'll just read the question. The substance produced by a cell in where cells from further infection into. Primary.
Which one? Is it the one innate immunity or acquired immunity?
Acquired one.
Innate.
Innate. Oh, okay.
Transplantation of tissue/organs to save certain patients often fail due to which is responsible for the graft rejection.
D.
Very good. CMI, which is mediated by which cells?
I didn't do this chapter. I just answered on that day.
It's okay. Antibodies present in colostrum.
B.
Tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component which causes this is
A. Nicotine.
Antivenom against snake poison contains which type of the following?
C.
Right. Which of the following is not a lymphoid tissue?
C.
Right. Which of the following gland is large size but reduces?
C.
Very good. Hemozoin is a?
C.
Very
Toxin.
good. All of the answers are same. Which of the following
Yeah,
is
this one
not
was
causal organism for ringworm?
C.
No, these three are. This is Microsporum, Trichophyton, Epidermophyton. These three are the fungi.
Oh, no. Sorry. I told 21. 22 is D and 23 is C. Yeah, 22 is D and 23 is C. Yeah.
Why?
Because the accumulation of cell itself happens because of its shape.
Because like this Plasmodium pathogen couldn't
Cloud.
enter. There's
Enter,
no
yeah.
space to do
Is
any
for
kind of
it.
thing. Okay, very good. All right, which chapter should we be starting today?
I don't know.
Shall I start biotechnology with you?
Okay.
That's a nice chapter. Okay, see this chapter, I will not be teaching just like that because we are almost nearing the end. Now all the chapters are EVS. Other than biotechnology, ecosystem is there. Then what is that other chapter? Organism and population is there. So these chapters are basically EVS. So, one more thing, biodiversity. Some of my notes are so nice. This is the second biotech chapter. You see the notes how colorful, colorful and
Yeah, ma'am, did you use that sparkly? Oh, yeah. Okay.
this is application. This is not what we are going to start today. We're going to start with the processes and stuff.
Okay.
This one is not as bright and shiny. This is also low. Where is the introduction part? Okay. So let's start with biotechnology. Now this chapter, I will not be taking it very quick. I'll take it slowly so that you don't get confused. And for this, you'll be writing notes. Okay, because this chapter, you cannot take it like this is very nice, but it can be tricky sometimes. So when we talk about biotechnology, I don't know why I've written after 5 p.m. So when we talk about biotechnology, it's basically a whole entire combination wherein there is plants, animals, microorganisms all into one. And then we are using certain human manipulating techniques to use the animals, plants, and microorganisms according to our own needs. Understood? So when we talk about the biotechnology, so the first chapter is principle and processes. So in this chapter, you're going to see the basic thing, like what is biotechnology, what do we do in it, and what are the tools for it. Like the basic introductory, whole entire chapter is introduction. After this comes the biotechnological application. Then you're going to see where is it actually used and how is it beneficial to us. It is indeed very, very beneficial. So when we talk about biotechnology, so it is the technology based on biology used in the field of agriculture, used in the field of food science, and in the field of medicine. In these three basic, you can say major departments, we can utilize the biotechnology and thereby reap a lot of, you can say, benefits. Now, there is a European Federation of Biotechnology that has given a very weird and so not nice definition of biotechnology. So, according to them, It is the integration of natural science and the organism, cells, and parts thereof, and molecular analogues for products and services. Trust me, I never learned this sentence back when I had 12, like this chapter in 12. Nor did I learn it for NEET, nor I know it now. If you ask me to, ma'am, read it without looking at it, I can't. So, see, basically what this weird sentence says is that we are taking the natural bioscience, that is plants, animals, and microorganisms, or we are taking their cells or parts of their cells, not the whole entire cell, but parts of their cells, or even their molecular structures, meaning you go at the gene level, you're going there also to make them or to use them for making our products and use them for our services. Understood? That is what biotechnology is according to ESB. Remember, the short forms are very important. They can give you anywhere to match anything. From random chapters also they'll give. So pay attention. Now, the principles of biotechnology. There are two principles of biotechnology. First is genetic engineering. Now here, genetic engineering, we are going at the molecular level, the genetics level. When we go at the genetic level, we have the nucleic acids in the body. We have the proteinaceous structure. Okay, so here, using the genetic engineering, we alter the chemistry of the genetic material. We attack the genetic material and alter it. Maybe we add something to it, or maybe we subtract something from it. But all in all, we alter in either of the cases. And then after altering, we introduce these into the host organism and then change the phenotype of host organism. I'll give you a very small, simple, crisp explanation to this. And then the last thing is bioprocess engineering is that when you're doing these things, when you're doing genetic engineering, when you're putting it into a host organism, so you need to maintain a very sterile condition, contamination-free area. You need to maintain that, or else things will not happen in your favor. Okay. So now, all in all, let me tell you, in this chapter, you're going to see that, for example, you want to produce like an organism that has superior characters. But, or you want to produce something in bulk, and you cannot do it with normal human manpower. Then you utilize other organisms for it. For example, the best example for it is human insulin. You know that those people that have diabetes, type 1 diabetes, they need insulin shots in order to manage their insulin level. Now, the production of insulin with the help of biotechnology is one of the most, you can say, successful work of biotechnology, wherein the human insulin gene is taken from human, introduced into bacteria. And then the bacteria is made, or you can say is allowed to make, like, you know that bacteria, they have such a fast rate of division. They divide within 20 minutes. So using their reproductive time and their capability of having this incorporation, because you cannot manipulate human genome. That's unethical, right? You cannot go to a person, inject them with something and then like, okay, you're my test organism. That's not going to happen. But bacteria, you can do on that. So using the bacteria, billions and billions of that human insulin is produced with lesser amount of money and better tech. Understood? So this is like one such overview for this. So here bacteria becomes the host organism. Understood? Okay. So these are the two principles of biotechnology. Now see, talking about this, now in this chapter, you'll have the mention of rDNA a lot. Recombinant DNA. See, this biotechnological chapter, this also revolves, you can also call this chapter as recombinant DNA technology, RDT, recombinant DNA technology. Now, what is the recombinant DNA? This is the DNA that has its, or that is altered. The altered DNA is called as a recombinant DNA. So these are the DNA molecules that are formed in the laboratory by using the methods of genetic recombination, such as cloning, that brings together genetic material from multiple sources. So that DNA which has been altered, either a lot of things has been added to it, or something has been subtracted from it. Okay, so that is called as recombinant DNA, which we are going to work with throughout this chapter. Clear?
Hmm. Hmm. Mom, so they use that for like changing the genetic of a child, like a baby or something like that?
Humans have never done anything on that.
Ah, okay.
Maybe the drugs that are developed after they have been tested and stuff, they are released into the market, then the human utilizes. Or maybe, let's just say, this thing, like therapies. Certain therapies are also given using recombinant DNA technology, but again, no testing could be done on humans. That is unethical, totally. Okay, all right. So this was the introductory part. Now coming on to the tools, the things that will be needed during this whole entire process. So first, we should know what is cloning. See, you know the clones are, for example, if this is a cell, several copies of this same cell will be called as the clones. These are clones of this particular cell, right? Exact copies, replicas. Okay, so the process wherein we make replicas, meaning several replicas of the initial template DNA. Template DNA means the DNA that we are doing the test upon. That process is called as cloning process. Understood?
Hmm.
So what are the steps in the cloning? First of all, you create a recombinant DNA molecule. How do we do that? You take the fragment of the DNA that is interested to you, like this is the gene of interest. You put it into a vector, which is a carrier, and then this together would become a recombinant DNA, the DNA that has been altered. Okay. Now,
Hmm.
after this has been done, like I told you that we put things into bacteria so that when the bacteria divides, we get that product from it. The same way, if you take this recombinant DNA, you add it into the bacterium and then the bacterium has that recombinant DNA in it. So this is the host, right? So we transport that recombinant DNA into the host cell. Now, when the host cell will divide, the particle of that recombinant DNA will also divide. Understood? So that will divide and later on it will be sent to the different generations as well. So, and this further will keep on happening and so on. Understood? Now, the first recombinant DNA, it was made by Stanley Cohen and Herbert Boyer in the year 1972. So, it's not a very old technique. It's very new. It's fresh in the market. Okay, 1972 is not that far away, right? So, this is when it was, you can see that it was made. Now, what they did is that they linked a gene that was coding antibiotic resistance. Antibiotic resistance means it is the property wherein the antibiotic will not work on that particular organism. That is called as antibiotic resistance. Resistance means to stop something. Okay, so they linked this antibiotic resistance with a native plasmid of Salmonella typhi. Okay, the one that causes typhoid. What is a plasmid? See, do you know what is a plasmid?
Where it's like we have plasma membrane, but then the prokaryotes have plasmid, which is like
So,
a copy.
बिल्कुल
Is
नहीं.
it?
Plasma membrane, prokaryotes also have plasma.
Yeah, no, but they don't have like a proper membrane sequence.
You're talking about nucleoid. I'm talking about
Ah, yeah,
plasma.
sorry, never mind, never mind. Sorry, sorry.
See,
Um,
in the
um,
time, try one more time.
plasmid. It's something which holds together, right? Like the things present in the cell,
Huh?
like something which holds together in the cell.
See, plasmid is the extra DNA that bacteria has. Like bacteria has its main
Oh
genomic
yeah.
DNA.
Sorry, sorry. I know that. Yeah, sorry, sorry.
Other than
I
that
know that.
main DNA, it has extra DNA.
Extra
And that is
DNA.
what
Yeah.
a plasmid is. So this plasmid has no connection with the main DNA. It is extra chromosomal DNA that the bacteria has. Understood?
11th grade I forgot. It's there.
So there are 3 basic steps in genetically modifying an organism. Okay, to modify the organism's genetics. First is one second. So creating a genetically modified organism, which is called as GMO. So for that, what do we need? Is that first of all, you need to find out the gene of interest. Whatever you want to incorporate into the organism, you want to find that out. So for example, first you're going to identify the DNA that has all the desirable genes that you need. Then you will take that identified DNA, put it into the host. Then you'll put it into the host and then you want to maintain it into the host so that the host further transmit it to the progeny. So that then only it is successful when it is carried, when it becomes the part of the host's DNA forever. Understood? So that is what we are going to do. Because if it gets lost in the middle, then it's of no use.
So they keep it in the DNA.
See, like you are introducing the recombinant DNA into the host DNA. Right? So you want that recombinant DNA to become the part of the host's genome. And thereby transmit it to the progeny. Then only multiple copies, meaning organisms with what you want will be prepared. So you want to do that for a long period of time. If you want a particular plant that has all the good characters, so you'd want all the progeny plants to also have it. So maintaining it is the biggest task. You do all of the processes, but to maintain it is very important. Is it clear?
Yes, ma'am.
Now we're going to see, like we saw how we are going to like locate the gene of interest. We're going to take it out. We're going to put it into the vector. We're going to put it into the host and all of that. But how are we going to do it? It's not all like this, and then. No, we need certain things. And it's not a it's not a macro level setup. It's a micro level setup. You can't do it just by hands. You need proper experimental things for it, right? Because you can never see DNA. You can never see a gene. You have to extract it. So let us see the tools of the recombinant DNA technology. First of all, there are certain enzymes that come into play, such as restriction enzyme, polymerase enzyme, ligase enzyme. 3 enzymes are there. Then we need a carrier. So that is our vector. And we also require a host organism in which you're going to incorporate all of these things, right? So that is what we need. These are the raw materials or the tools without which the recombinant DNA technology is not at all possible. So we're going to use all of these things. The first one is restriction enzyme. But first, you note down here.
Ma'am, can you scroll down? 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 Ma'am,
Hmm.
I wrote the heading.
You wrote the heading this one.
Yeah.
Ah,
Is
you
it
write.
okay if I
Ah,
continue
you
in
write
this
it under
one?
it only. Ha.
Okay. Ma'am, can you scroll down? 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 Ma'am, can you scroll down?